So good morning. First, I would like to thank the speakers from this morning. It's absolutely wonderful to see all the initiatives and projects that are happening within CGC, and I really hope this is inspiring to all of you to join some of those projects and take an active role. But what I thought I would do in the next 10-15 minutes is to introduce efforts that are happening related to cancer curation within different group, within different entity, which is Clinical Genome Resource, or ClinGen. And I thought that it would be very important to introduce this effort to CGC audience, because I'm sure there is a lot of opportunities to overlap, coordinate, harmonize between groups and collaborate, and that we will see some overlapping goals. So I'm presenting on behalf of the entire Cancer Curation Working Group within ClinGen. I'm sure you are familiar with ClinGen, that you have seen this website multiple times, but just very briefly, it's an NIH-funded effort, and the broad goal is to develop knowledge and standards and resources for a clinical interpretation of genetic and genomic data for better patient care. So some of the key activities are to support data sharing between patients, laboratories, and clinicians, to develop standards and guidelines for clinical interpretation of genetic and genomic data, to perform curation. Some of the curation activities are listed here: gene disease validity curation, dosage sensitivity, variant pathogenicity, clinical actionability. So basically, all the information and resources and processes that you may need to answer key questions, whether a gene is associated with disease, or is a certain variant causative for a phenotype, or is it this information clinically actionable? That's what ClinGen tries to provide. So a related entity is ClinVar. Those are two separate entities that are supported by NIH, but very closely related and working together. So a lot of the ClinGen activities are actually supportive to ClinVar. So ClinVar is a very big database that aggregates genetic genomic variants together with information about their relationship with disease. And we all know that ClinVar is absolutely invaluable resource for all of us in our daily work when we try to interpret germline variants. Somatic variant interpretation, as we know, is just as massive and daunting task as interpretation of germline variants. And I'm sure you remember this very nice figure from a talk yesterday. It was an excellent talk by Alex Wagner, who is our travel awardee, showing how it's actually a curation and interpretation part, the bottleneck in performing somatic genetic testing. As we are getting better in performing sequencing itself and then primary data analysis, it's really the toughest part becomes to interpret clinical significance of somatic variants. So one of the ways that we are addressing this as a community is generation of public knowledge bases that catalog clinically relevant cancer genes and variants together with interpretations of their clinical significance and supporting evidence. Some of these knowledge bases are listed here, and although entering information to knowledge bases is still manual, requires manual curation, by generating these shared these knowledge bases, we're transforming that into collective effort, so that we are not repeating this work within our laboratories, but we are sharing this information through open knowledge bases. And so there is certainly a lot of work involved and a lot of work for everybody. So where does ClinGen fit into this space? So several years ago, an initiative was started within ClinGen to apply some of the knowledge and expertise and resources that exist within ClinGen and were applied to curation of germline constitutional variants, and so to apply some of this to the problem of somatic variant curation. So Somatic Working Group was formed, and it is co-chaired by Shashi Kulkarni from Baylor and Subha Madhavan from Georgetown, and we have more than 60 members by now from a variety of different academic institutions and commercial partners. This is a photograph from a last face-to-face meeting that was at AACR meeting in Chicago this year. So some of the key milestones and accomplishments of this working group to date. In 2016, the group published an article describing MVLD standard. That stands for Minimal Variant Level Data Standard. And if you're not familiar, I will describe that in detail in the next couple of slides. There were also a couple of other articles describing initiatives within ClinGen's Somatic Working Group. I especially want to bring to your attention upcoming human mutation special issue that will have one of the articles dedicated to collaboration between Somatic Working Group and Civic knowledge base. The working group has selected Civic as a curation platform, so we have very close collaboration with Civic. Curation task forces were formed to focus on curation of variants within different tumor types or for different cancer genes. We have recruited curators that work with different task forces, but we are still looking for additional curators. And we're actively adding genes, variants, and evidence items into Civic. Code has been written to transform information from Civic into ClinVar for broader distribution of information. And then we are constantly working on improving the process of curation and making it more efficient. So back to minimum variant level data. If you remember, again, from Alex's talk, he very nicely described the problem that we are having because of a lack of standard and standardization and harmonization how somatic variants are presented in different resources. In this case, concretely, in different knowledge bases. When WIC consortium tried to network and harmonize between different knowledge bases, this became a big issue that everybody is using different way of presenting somatic variants and data related to somatic variants. So this is a problem that ClinGen Somatic Working Group realized early on, and this publication about MVLD was an attempt to address this issue. So what MVLD is, it's a proposed standard for minimum variant data that we should be presenting for somatic variants and standard ways of presenting each of these data variants. So for example, in MVLD standard, the data is grouped into allele descriptive information, allele interpretive, and the somatic interpretive. So allele descriptive is basic genomic information about that variant, what is the genome build, what is chromosome, genomic coordinate. Then allele interpretive goes more in depth about that allele and its effects on cDNA level, protein level, functional level for that protein. And then somatic interpretive is really information about the significance of that variant for a particular tumor. So this includes information about which tumor type is the variant associated with it, what is the biomarker class, is this variant diagnostic, prognostic, predictive, is there any therapeutic association, what is the effect of the variant, is the variant predicting resistance or sensitivity. Very importantly, we need to standardize level of evidence. So MVLD proposed one model for standardization classification of evidence, but in the meantime, ASCO-CAP guidelines came out that proposed a system for evidence classification and variant classification. So now MVLD is incorporating information, the system, classification system from these guidelines. So although MVLD was a very good first step, there is a lot of effort that needs to continue in terms of standardization and harmonization. For example, we do not have agreement how should we even be classifying tumors, what classification system or approach when we are representing somatic variants. Should we be using OncoTree that's becoming popular with some knowledge bases? Should we be using human disease ontology? And there are other things that still also require standardization and common drug databases, standard terms for conveying variant effect, and so on. So this is an ongoing effort, and hopefully we will eventually be able to develop standard that will be very broadly accepted. I already mentioned that we have close collaboration with Civic, which has been selected as a curation platform for our efforts. So this is just showing ClinGen as one of the groups that are associated with Civic. And then formation of different task forces really helped to focus on specific tasks and move those projects forward. So these are task forces that exist within the working group to date. And the group or subgroup or task force that focuses on curation process itself, trying to find ways to make it more effective and streamline it. There's Pediatric Task Force, where I belong, and that was what inspired me to join Somatic working group efforts, because I I really wanted to get engaged in this effort. There's Pancreatic Task Force, Non-Small Cell Lung Cancer Task Force, TP50 Task Force, and a subgroup that works on continuing harmonization and standardization. And of course, with time, we will probably be generating additional task forces and expanding these efforts. In terms of the curation process, what seems to be shaping up as our approach, and of course with close collaboration with Civic, is that somatic task forces are starting to act as expert review groups for Civic, for curation in Civic. So as you know, Civic is crowdsourced as curation, so everybody can contribute to Civic, but then there is expert review of all the data that's entered in Civic. So task forces are serving more and more as these expert review groups. So this is a model that we use, for example, in somatic in pediatric task force. So we have a group within pediatric task force of experts. We prioritize tumor types, genes and variants for curation based on clinical relevance, based on lack of data and knowledge bases about particular genes and variants. And then curators start gathering publications, evidence items, and generating records in Civic with associated PMIDs, associated publications. Once there is a sufficient series of evidence about a particular variant or a gene, that gets reviewed, and we decide whether there is sufficient evidence to create assertion, which is a quick summary about clinical relevance about a particular gene or variant supported by evidence. And then those assertions are additionally reviewed and approved by task force members. And then the work group is constantly working on finding ways to make somatic variant prioritization and curation process more effective, to have mechanisms for multiple curator reviews, and to also scale the efforts, so engage a broader community, and especially trying to engage trainees in these curation efforts. So just brief overview of different specific task forces. As I mentioned, I'm part of Pediatric Task Force. And so this was started it's because of realization that although all pediatric cancers have completely different genetic and genomic composition than adult cancers, there is very little information in knowledge bases to date that focuses specific to pediatric genes and variants. So far, the task force has prioritized a list of 44 genes and variants and tumor types for curation. We have monthly conference calls, and these conference calls are focusing on specific group of variants or specific gene or specific tumor. So for example, the most recent one focused on NTRK fusions. So the genes and variants are in the process of being curated from our priority list. We also work with Civic to enhance search of information that's pediatric cancer specific, so some kind of flagging so that you can specifically search for pediatric relevant information, and we are starting pilots to involve our trainees in this curation process. There is also Pancreatic Taskforce, focusing on pancreatic adenocarcinoma, which is a really aggressive, deadly disease without effective biomarkers for early detection. So they use PanCan and known tumor resource to prioritize genes and variants. There is non-small cell lung cancer tumor, again, a common tumor type with high clinical significance and with therapeutic associations. So it was felt that it's very important to have highly curated and high quality information broadly available for this tumor type. TP53 Somatic Taskforce, which was formed because of the role of TP53 into so many different tumor types, and then challenges when it comes to interpretation of TP53 variants that can be tricky, especially point mutations, and to interpret their oncogenicity can be hard. Somatic TP53 Task Force works very closely with Germline Expert Group on the same gene. And then in addition to all the work that's happening within specific task forces, as an entire working group, we continue to think about harmonizing existing guidelines and guideline efforts and curation efforts between different stakeholders and groups, including ACMG and PASCO. So hopefully we will be all able to agree upon collectively shared and supported standards in this area. Constantly thinking about increasing efficiency of a curation process and increasing usability of all the resources that we are developing for physicians, patients, obviously laboratory professionals like our group here, and payers and regulatory agencies. So with that, I just want to thank, again, everybody in Somatic Working Group, Suba and Shashi, all the chairs of individual task forces, wonderful ClinGen scientist, Deb Ritter, coordinator Meredith, and then everybody in Civic team they're absolutely wonderful to work with. Very supportive. And then in addition to being active in CGC, I also want to invite you to consider if this is something that could be of interest to you, to consider maybe getting involved in this type of work.